epigenome editing, we should know that why we should go for epigenome editing, isn't it? So, what is the role of epigenetics and for what purpose or what are the characteristics for which we can go for genome editing, so that we can improve our crop plants. Okay. So, let us see how can we define genome editing. Okay. So, we have been doing by making genes for transformation, all editing we have been doing in vitro. Okay. So, here also we are doing some engineering in vitro and then allowing other things to happen inside in vivo engineering to happen. In case of transgenic, most of the things, most of the engineering has been being done in vitro. So, here we have to do something part of that in vitro and then remaining things editing will be done in vivo. So, this is one of the recent techniques, because this was proposed or invented, we can say somewhere in 2012 by Jennifer and her group, and they found that there is similar thing happening in bacterial system. So, they used that system and thought, why not, can we use that in animal system or plant system? Initially it came in animal system, now we are trying to use them, we use that technique for editing plant genome. So, what is specific here? Whenever we are doing transformation type of things, that is random process, where you want to insert your gene or DNA or transgene, that is not in your hands. Here we want to do editing or changes, either incorporation of the transgene or making some base changes, minor insertion, deletion, all sort of things at our will, at a specific site, particular site, wherever we want to do. So, that is how this is targeted genetic change in the genome or gene. We can say we can target one gene, because that is, or maybe several gene at a time. So, those things we are going to see, either by adding, you can add, you can remove or you can replace. So, all sort of things can be done using genome editing. And of course, so many countries they are not having regulatory process, whether this should be considered as genetically modified manipulated organisms or not, that is being developed, but overall it is considered that that is safer or should have lesser biosafety concerns compared to the transgenic plants. So, that is how we can say developing transgenics using genome editing technique will be having less concerns compared to the development of transgenic plants using the conventional. Conventional means you have to use selectable marker, scoreable marker, vectors. Either without vector you are going to transfer your gene, that will be clean DNA transformation, or with the vector. If with the vector it is going inside, more concern will be there. Okay. So, there are certain requirements for genome editing, and so far at least 4 or 5 different methods have been known for doing genome editing. So, use of mega nucleases. So, what we need, there should be protein or some other molecule should be there, maybe RNA, which should recognize your site of interest, where to do the editing, and then other things should be there that should cut the DNA. So, 2 components. So, mega bases also almost all these 4 things contains 2 important things. Little bit we will discuss, and we will also discuss about advantages or disadvantages of each of these 4 things. So, use of mega bases, mega nucleases, and then zinc finger nuclease can also be used, and then talons, and then the most recent one is CRISPR-Cas system, and then we will also see what are the recent advances in CRISPR-Cas system. So, in case of mega nuclease, again you can see that there is fusion of 2 protein. Okay. So, one protein is going to identify the binding site, and that is the site of action. Okay. Another protein is fused with that, that is going to cut the DNA. So, both together, and then wherever one protein is going to bind, next one is going to cut the DNA. Okay. So, it is not that you will always like to have editing at one place. In the genome, anywhere you may wish to have editing. So, binding site has to be changed, means you need to manipulate the protein which is going to bind to the DNA, and in that way it is very difficult to do protein engineering. You are going back to protein engineering, so that you are going to change that binding protein, and then only, and that nucleus will be attached to it. So, wherever that protein is going to bind, nucleus will make a cut. So, you have to do protein engineering, then only it will be successful. So, very difficult to frequently change the site of action of this nuclease. So, that is one limitation of this technique. That is why it was little bit difficult to do that, means use it for genome editing. So, next one is zinc finger nuclease. So, here zinc fingers, that structure is there which is going to bind to the DNA. So, that is going to recognize the binding site, and nuclease is attached to it. So, nuclease will make a cut. Okay. So, again you have to manipulate the zinc finger, so that you can change your binding site, and then nuclease always attached with that. So, it is going to make a cut. So, again difficulty arises because of you need to manipulate the binding site of that particular zinc finger. Okay. So, similar difficulty here also. Then talons are there. Okay. Talons can also be used. So, here again there is a stretch, and around 30 nucleotides, and among this sequence, two bases are very important, two nucleotides are very important, 12th and 13th, that is going to recognize binding site of that particular talon. Okay. And once it is bound there, then nuclease is already attached to it, it will make a cut. So, similar problem here also, you have to make manipulation and changes. So, that will create some hindrance in manipulating the DNA of your wish, okay, or the DNA or gene of your interest. Then CRISPR-Cas, the last one we are going to discuss is the CRISPR-Cas. Here again there are two components. One component is going to bind or identify the target site, and attached Cas, that is CRISPR associated nuclease, that will make cut. Okay. So, if we see, there are not protein involved here, RNA and nuclease is protein. So, RNA and protein, they are involved together, and if we look at the RNA part, there are two naturally occurring CRISPR. If we talk about, there are two different types of RNA involved, that is responsible for identifying the binding site, and then associated Cas, that nuclease will make a cut. Okay. And later, while doing research work, the people find that if we can make both hybrid of that RNA, single RNA, so that will also work. So, it will be, instead of handling with two RNA, if you are going to deal with only one RNA, that will be little bit easier, and this CRISPR-Cas is easiest than whatever we have discussed earlier 4 systems. Okay. So, if you want to manipulate or synthesize RNA, that will be easier than synthesizing protein, and that too very small part of the RNA molecule. So, that single RNA molecule, that is guiding where to go that nuclease and bind. So, that is why that is called guide RNA, guiding the enzyme to bind and then act upon the DNA. So, single, it has been from two RNA molecules, it has been converted into single. So, single guide RNA, sgRNA. So, whenever we want to do some editing, you have to find out what place to make do editing, and then according to the sequence where that you want to edit, you have to prepare guide RNA, and that guide RNA will be attached to the nuclease, that is CRISPR associated nuclease. So, that will be associated with that, and wherever complementary sequence is there, guide RNA will go and bind, and then it will Cas nuclease will make cut on it. So, you can see here, this is the 4 steps how the action is action of CRISPR Cas is happening. So, these are the 2 components, first component and second component, and this is the nuclease, and here that RNA will be bind together. So, this is one complex. So, this RNA is going to find out the place where to bind, and then you can see here. So, this is going to find out the complementary sequence, and once complementary sequence, of course, we will see that about this RNA. Then nuclease, this is the nuclease, this will make double stranded cut. Okay. So, this is that gene or genome is now cut into 2 parts. Okay. And you must also be knowing that there is a repair system in all living organism. DNA damage, if a damage is happening, repair system is there. So, this cut, so it will appear like that, double stranded cut, 2 different fragments, then it has to be joined together by repair system. So, there are 2 different repair system. So, when joining will be there, randomly there will be insertion of any base, whatever base is available, that may be inserted. So, you can see that there is some mutation. Okay. So, because of mutation, and you must be knowing that there is codons, coding sequence is there. If one single base is getting inserted, the reading frame will change, and the nature of protein will change, and of course, that you are going to target particular site, which is very, very important for the activity of that enzyme or protein. So, there only you are making changes, and because of that change, protein will not be active or it will not work. So, you will find that that particular trait or trait associated with production of that protein, that will disappear, that will not be there. So, you will find the mutant organism, plant or whatever it is. So, this sort of insertion, or if you are going for homologous recombination, you are providing similar sequences, then your gene or stretch of DNA may get inserted at this place, this particular place. So, that is how you can generate transgenic plant also using this CRISPR-Cas technology. So, you have to go for different study, either you want random base to be inserted, or you want your gene of interest to be incorporated by homologous recombination process. Okay. So, this is in brief about CRISPR-Cas technology. So, let us see the recent advantage. Of course, this is this technology is not more than 7 years old, but there are lot of advancement happening in that. So, we have seen CRISPR-Cas, and here you can see D-Cas. Okay. So, this D has been added because of dead Cas, means that Cas is not dead, it is not able to do its own function. And what is that function? Nuclease activity. So, if you remove nuclease activity, what will happen? So, this RNA, guide RNA will go and identify the place where to bind, but nuclease is not able to make a cut. Okay. So, you can replace nuclease with any other enzyme, and what type of enzyme will be there, that is what we are going to see in after seeing that epigenetic and epigenome editing. So, this type of construct, they are not going to make a cut, but they can be used for regulating gene expression. So, you can, instead of Cas, you can add some protein which is going to either boost the expression of that gene or slow down the expression of gene. So, you can manipulate, regulate level of expression of gene. Okay. And this is not going to make a cut because the Cas is dead, not active. Then CRISPR-Cas12. Okay. So, later on it was found that there are certain nucleases which are very, very specific and doing very effective work. So, that was named, of course, in the sequence, that is Cas12. So, very effective, more effective than Cas9. Okay. So, if you want, there should not be any erroneous manipulation or editing, then you can use Cas12, very precise and very effective. Okay. Then subsequently another nucleus, that is Cas13 was identified, and very peculiar things about this Cas13b is that is able to edit RNA, and you know RNA is single stranded and even single stranded DNA can be there, but Cas13b identifies RNA and it single means it needs to make single cut, cut in only one strand, because RNA is having only one strand. Then subsequently Cas14 was also identified. So, substrate for Cas14 and Cas19, how it is different? Cas19 requires double stranded DNA, Cas14 single stranded DNA, and Cas14 and Cas13b can identify that this is single stranded RNA and this is single stranded DNA. So, they will bind to 14 will bind to single stranded DNA and 13b will bind to single stranded RNA. So, whenever we need to manipulate or need to do editing in, say for example, transcriptome editing, if you want to do, we can use Cas13b. And then you must also be knowing that there are certain viruses which are having single stranded DNA as genetic material. So, if you want to do manipulation or editing there we can use Cas14, okay, not Cas9 or Cas12. So, further advancement and identification of particular Cas, the Cas was identified which can be reversibly and inducible nature. So, whenever you provide inducer, it will go and bind to the target site. If you remove the inducer, it will come back. So, reversible and inducible nature of that Cas. So, whenever you want, okay, do the editing, it will do. If you are okay, stop it, it will not do. Maybe the next generation what you want to see. So, you provide the inducer, in the next generation also it will do the editing. So, something like that. So, that was all about of course, in brief CRISPR system or gene editing tools, different tools. And you must be knowing that what is genome and how do we define genes. Gene or genome, if we talk about gene, that is nothing but nucleotide sequence, okay, which codes for a functional product. That product may not be only protein, that may be RNA molecule also. So, whether it is RNA molecule or protein, that is if it is coding for that, then it is the gene. And if you remember little bit about the genetics, you must also be knowing that mutation. What do you mean by mutation? Any change in nucleotide sequence. So, that comes under genetics. So, there should be change in the nucleotide sequence. If change is there, that is the domain of genetics. If without, and if mutation is there, you know that mutant, some mutant is there, a particular trait or the gene associated for that trait will disappear. Okay? So, mutant is not having that particular trait. So, this genetics is dealing with whether the expression of gene and not only expression of gene, transfer of genetic material from one generation to another generation. Let us see what is genetics. By name itself, no, EP plus genetics. So, something is added, and EP, if you see the Greek, this is a prefix taken from Greek language. So, that means something from outside, external. Okay. So, the changes happening from the outside or because of external things. Okay. But the definition is that there should not be any change in nucleotide sequence. If change in nucleotide sequence is there, then it is the domain of genetics. So, here no change in the nucleotide sequence, but expression of the gene should change, and we have seen that expression of gene changing if some mutation is there, means change in nucleotide sequence. Here no change in the nucleotide sequence, but gene expression is going to change, either reduced, increased or disappear, whatever it may be. And we find that there are so many traits and even the developmental processes in all the living organisms, whether it is animal system or plant system, they are regulated through epigenetic changes. And this was not earlier known. You may remember Lamarckism. Okay. So, the law given by Lamarck was inheritance of acquired characters, and nobody, and after some time Darwin proposed the law natural selection or survival of the fittest. Okay. And if you see, if you remember, law of Darwin, whatever he proposed, that was well accepted, and everybody started saying no, Lamarck was not, means whatever proposed by Lamarck, that is not true, because he could not give evidence of inheritance of acquired characters. And you may also remember what was his experiment, cutting tail of mouse for several generations, and he found that, or people found that newborn mice, they are all having tail, even their parents were not having tail, because in mice you will find that you cut the tail, after some time again it will grow. So, they were continuously cutting it, and male and female having no tail, that was producing progeny with tails. So, that is how people were not ready to accept it. And then whatever evidence given by Darwin, that was well accepted. So, today it is not that Darwin's theory is not, means has become false, it is not like that, that is true, but the changes because of epigenetics or knowledge of epigenetics is that we have to accept whatever was proposed by Lamarck. And the Lamarck said that environment can affect your gene expression. Indirectly, this was the, or environment can affect your, in older words we can say genome, but now we should say environment can affect your epigenome. Okay. So, that is what we are going to see, and we find that if we look at human being, how many genes are there in human genome? Around 30,000. Okay. And do you think that all the 30,000 genes are expressing always, from the morning also, or maybe childhood also, teenage also, and young stage also, and then old stage? No. Similarly, in plants, when plant is going to germinate, all the genes are not expressing. At vegetative growth also, all the genes are not expressing. Reproductive also, genes are not. So, those, say for example, genes responsible for making flower, why they should express at early stage or at the time of germination? So, only those genes should express which are required, and the genes expressing in root, they are not expressing in shoot. So, there are so many regulatory processes which are affecting expression of gene. So, one of them, now very well known, one of them is epigenetic changes. Okay. And so, epigenetic changes are happening and regulating expression of gene throughout the life of the organism, and not only through the developmental stages, also at the time of environmental stresses. And environmental stresses may be of any kind, maybe heat stress, maybe drought stress, maybe starvation of particular nutrient, or maybe you are shifting to one type of food to another type of food. Say for example, you are every day eating rice and chapatis, but after some time, maybe after 2 years, 3 years, or maybe after your 5 year of age, or maybe your kids, that is shifting to pizza and burger. So, that sort of changes will make change in the epigenome, and that consequences you can see, or people are seeing. That also I will try to explain little bit. So, what are the different types of epigenetic changes? Okay. So, epigenetic changes, of course, there are 3 important components. The first component is DNA, changes in DNA. Okay. So, different type of changes are known, and the first one, okay, before that let me ask you, how many bases are there in DNA? 4 bases are there, everybody, all of us know that. If I ask you what is the 5th base, then what will be your answer? 6th base, 7th base. We are very, you should be at least after this training program, that 5th base is methyl cytosine. Okay. Cytosine, normal cytosine, where methyl group is added at 5th carbon position. Okay. So, that is called 5th base of DNA. So, I said 6th, 7th, all those things are there. So, another modification, 5-methyl cytosine is getting further methylated, modified with addition of hydroxyl group. So, 5-hydroxyl methyl cytosine, hydroxymethyl cytosine again at 5th position. So, that is called 6th base. So, similarly, this is very well known. Of course, the number of hydroxymethyl cytosine is very, very less, number of methyl cytosine is very, very high. Similarly, there are other modifications also, say for example, 6-methyl adenine. Okay. So, that is also appearing, and all these modifications are very well known in animal system. And after knowing all these things in animal system, we are now trying to see whether they are present in plant system also, and if they are present, how they are going to affect or regulate gene expression. And if we know that, what is our target? So, regulate expression of gene under particular circumstances, maybe some stress or maybe normal growth condition, but we want to this particular trait should, or this particular gene should express, over express. So, for that reason only we are studying this. And if you are not aware, diabetes and cancer, these 2 diseases are very, very spreading very, very fast, and the reason behind it, there are several reasons being said, but epigenetic changes is the cause of that. Okay. And I said, if you are going to change your diet, so how your diet is going to affect your epigenome, and that is happening. You eat simple wheat and rice, means cooked normally, whatever we cook in house, or you eat the same product made of rice and wheat, but junk food like pizza and burger, and you must be aware that there are a lot of chemicals being put in that pizza and burger. So, those chemicals, they are going to affect your epigenome, and you will find that there are markers identified, well known markers. By checking the epigenetic changes, the doctor can suggest that maybe after 5 years or maybe after 10 years, or you are having cancer. So, that is possible, and now people are thinking of epigenetic treatment of cancer. So, that may possible, if not next year, maybe after 5 years or maybe after 10 years. So, we have to see how much perfect we are going to be in next few years, and then that may come in the picture. So, one change was changes in DNA, particularly methylation of cytosine, and then other changes also I said. There are 4 bases, I have talked about only 2 bases, adenine and cytosine. Guanine and thymine, those changes are not yet known. So, maybe in the coming days or maybe coming years, we can know changes are there in adenine and guanine, thymine and guanine also. So, that we have to see. And what is the problem? They are so less frequent that we need technology, some technology to identify, and that technology should be very sensitive. So, even out of 1 billion single base is modified, that we should be able to identify. Why we are able to identify methyl cytosine? Because they are very huge in number, that we will see. Okay. So, next type of change is in protein, and you know that histone proteins, nucleosome structure, in that DNA is wrapped around the histone protein, octamer histone protein, and there are 4 different types of proteins, 2A, 2B, H3 and H4. Okay. H1 is also there, but that is not the part of octamer. Okay. And in that octamer DNA is wrapped around. Okay. So, the histone is made of protein, and protein is made of different amino acids, and you know there are 20 different amino acids. So, those amino acids can get modified. Say for example, you must be knowing lysine is one of the amino acid. So, lysine may get methylated or it may get acetylated. Okay. So, it will have different properties than normal lysine or normal any other amino acid. Okay. So, it will have that effect, and what is found that if lysine is going to be acetylated, okay, so binding of DNA on that protein will be loose. So, it can easily open up. If lysine is going to be methylated, then methylated or modified DNA or modified protein, they may make very compact position. So, DNA, wrapped DNA wants to unwrap, it will be difficult, and you know that for expression of gene, that DNA has to unwrap, so that transcription can take place. When transcription is over, again it has to come back and wrap on the histone proteins. So, that is how modification in protein and modification in DNA, they are going to interact with each other. Even one of them, either modification in DNA, wrapping will be tight, or either modification of DNA, wrapping will be tight, or if both are there, both can interact there, means they cannot be separated. So, something like that, holding to each other, 2 different DNA and protein holding together. So, unwrapping is not there, expression will not be there. That is how they are going to. Okay. Third component is a small RNA, like we have seen there, no, guide RNA. What was the role of guide RNA? Finding out the place where to bind. So, similarly, so all this modification of methylation of cytosine, it will not happen randomly, something is required. What is that? Enzyme, methyl transferase enzyme is required for modification of cytosine into 5-methyl cytosine. Okay. And then do you think that there is only one cytosine in the genome? There are so many cytosines. So, which cytosine to be methylated? 2, and even you consider one gene maybe of 1 kb, you will find 250 cytosines. So, which cytosine in a gene to be methylated, so that gene should stop or increase its transcription. Okay. So, this guide RNA is going to find out the place where this methyl transferase will bind, and it will do its methylation of cytosine. So, which cytosine to be methylated is decided by a small RNA. So, that is how a small RNA is also playing a role in epigenetic modification. Okay. If a small RNA is not there, then methyl transferase enzyme will not go and bind, and it will do its action. So, and if more methyl small RNA is there, it will bind with methyl transferase, and so many wherever complementary sequences are there, methylation will be done. So, biogenesis of a small RNA more or less is also going to affect methylation process. Okay. And similarly, methylation of DNA also, histone protein also, enzymes are required. Okay. So, you can see here, so this is cytosine, and how this cytosine is getting converted into methyl cytosine, and then methyl group is added at 5th carbon position, and then you can see SAM, you must be S adenosyl methionine. So, that is going to provide methyl group for attachment at the 5th carbon position. Okay. And then you can see methyl transferase, DNA methyl transferase enzyme is involved in this process. Okay. And if we look at what is the occurrence or how much cytosins are methylated in different organisms, you can find that in case of insects 0 to 3%, maximum 3% cytosins may be methylated. So, similarly, you can see different vertebrates, it may be 2 to 7% cytosins are methylated, not all, only 2 to 7% cytosins will be methylated, and that may be involved in regulation of gene expression either during developmental stage or under particular stressful condition. So, if you look at fish and amphibian genomes, you will find that about 10% of the cytosins are methylated, but if you look at the plant, different plants, you may find in some particular plant more than 30% cytosins are methylated. So, this says that cytosins are really 5th base. It is not randomly sometime methyl cytosine comes, so much methyl cytosine. So, it appears that there is 5th base occurrence is so much. So, that otherwise out of 100 you will find 25% of each base. So, of course, out of 25% 30% are methyl cytosine. Okay. So, they are very much abundant, not very less. Of course, in some organisms they are not there. Okay. So, if methylation is happening, I said, and generally what is the perception, of course, the site of methylation also vary, that I will say tell you later. So, if DNA is going to be methylated, expression level of gene, the common or general perception is that gene expression level will come down. Okay. So, whenever expression of this particular gene is not required, methylate the cytosine, some of the cytosines should be methylated, and after 1 day, 2 days, or maybe after some time, that gene has to again express. So, what to do? That methylation should be removed, isn't it? So, some enzyme should come and should remove the methylation. Okay. So, the again enzyme is demethylase. Okay. So, methylation of DNA is a reversible process. You can see this methyl cytosine going to convert it into normal cytosine. Okay. So, this process is called demethylase. Now, coming to histone modifications, different proteins I have already told, and there are enzymes involved in modification of histone protein. Okay. So, if histone protein is methylated or modified, it has to be come back to the original position. So, demethylases are also there. Okay. So, both the process methylation or demethylation in case of DNA and in case of protein, they are regulated by enzymes. Okay. And then as I said earlier, where to do methylation, either in case of DNA or in case of RNA, that has to be guided by a small RNA, and in this case, in particular case, we call it siRNA. Okay. Okay. So, these are three components: a small RNA, these are DNA wrapped around, DNA wrapped around, and these are histone proteins. Okay. So, what happens? Tail of the histone proteins means last part of the histone proteins, and amino acids present there, they are getting modified. Say for example, these are the tails, and they are protruding out, coming out like hairs, no. So, these are all tails of the histone proteins, and at the tails, no, they are going to be modified. So, this ball type of a structure, no, this is not modified, this is modified. Okay. And these modified tails are identified by certain protein. Okay. So, if this protein is binding these two, it will be very difficult for this particular part of the gene to unwrap for transcription. So, additional binding element. Okay. So, you can see here this is the tail and there different amino acids are getting modified. Okay. And then there are remodulator proteins which will bind to this linker DNA, you know, two nucleomes in between whatever DNA is there, that is called linker DNA, and once this is binding here, this will ask the nucleome to move, means making this particular DNA free for transcription. So, this is going to help in transcription process. So, they are all going to help in expression of gene. Okay. So, I have already mentioned non-coding RNA. So, if they are available, they will allow histone protein or the enzymes involved in modification of histone or modification of RNA or for modification or demodification, means coming back. Unless these small RNAs are not available, that action of enzyme will not be there. So, indirectly they are helping in modification process, and that is how they are going to regulate gene expression. So, you must be aware of this picture, the story I have already told you. And what happened? This was explained in different ways. Mendel could not, Lamarck could not explain what was that, but today we can explain. And that day Lamarck could not coin the term epigenomics or epigenetics, but today at least we have coined the term, and we find that there is direct effect of environment on our genetic material, or we can say epigenome or regulation expression of level of expression of the gene. So, you must also have seen that particular gene is expressing more in particular tissue, less in particular tissue. So, how that or at a given time this gene is expressing, and immediately after next second or change in the environment, the gene has to stop. So, such quick or effective switching on and switching off the gene is very difficult if we not include epigenetics in that picture. If you include epigenetics as one of the regulators of gene expression, this is not only the regulator, but this is one of the regulators of gene expression, then it will be very easy to explain how genes are immediately switched on and how genes are immediately switched off, because of methylation or demethylation. Methylation stop the expression of gene, demethylation again start the expression of gene. And you must also be aware that there are different parts of a gene: promoter, coding region, and then terminator. So, transcription starts from the coding sequence and it ends just before the terminator part. Okay. So, if methylation, the general perception is that if methylation of a gene will repress its expression. So, that is related to methylation of promoter. If promoter C cytosines at promoter is going to be methylated, if methylation is happening, what is the site of means what is the use of promoter or why promoter is there? Promoter is the binding site where RNA polymerase is going to bind, and then it will start transcription from translation start site, isn't it? So, if methylation is happening, that RNA polymerase will not be able to bind at promoter, or binding will be very inefficient. So, less transcription or no transcription. If methylation is removed, polymerase, RNA polymerase will go and bind, transcription will start. So, that is how, and this is almost common thing. But if methylation is happening in coding region, so the effect of methylation may be anything. It may overexpress or repress expression of the gene. So, that you have to check in which gene what is happening, whether it methylation is overexpressing or repressing the gene expression. So, there we do not know, we have to study one by one, and then when you can say okay, methylation and this part of methylation. And then again, because many of you are from bioinformatics, so whenever you are okay, you might be using or you will be using genome sequence, okay, and sequencing the genome. You may get the data, you have to analyze and tell so many stories about what is there in the genome, where is promoter and all those things. So, now and we are doing that epigenome or epigenetic sequencing, epigenomes. What is difference in that? If you go for normal Sanger sequencing or any normal sequencing, you will find only ATGC, you will not find methyl cytosine, means you are not knowing anything about the epigenome. Of course, those things are there, methylated cytosine is there, but your technology, your sequencer is not able to differentiate between cytosine and methyl cytosine. So, you have to use such method. Say for example, do you know any sequencing platform where methylated cytosine can be known? You might have heard of nanopore. So, nanopore sequencer can, you have to train that machine, or even SMRT you must be knowing. So, if you train that machine for that, you have to tell that this is cytosine, this is methyl cytosine, and wherever this of course difference will be there, no, one methyl group is added. So, you ask the machine to identify and tell me the difference, machine will tell you. If you do not, that is what machine learning, you have to train the machine should learn how to identify. Okay. So, that is why people were not knowing normal Sanger sequencing and Illumina and this and that platform, they are not going to differentiate between cytosine, methyl cytosine or adenine or methyl adenine, no difference. But by bisulfite modification, another sequencing technology is there, in which what happens, you have to give bisulfite treatment. Bisulfite, one chemical compound is there. If you put your DNA with bisulfite, what will happen? All the methylated cytosine will remain like that, unmethylated cytosine, normal cytosine will get converted, demethylated. So, it will get converted into T. So, if you remove means if you treat your normal cytosine with bisulfite, it will convert into thymine. Okay. So, all non-methylated cytosine will convert into thymine, and so many thymine will be there, and you have to do next step is doing PCR. No, sorry, not thymine, it will get converted into uracil first. Okay. And when you are doing PCR, that uracil, what is complementary to uracil? Thymine. So, at the place of thymine, uracil, thymine will come. So, in your sequence read, you will find that there are so many thymine, because thymine was already there, and then cytosine, normal cytosine got converted initially to uracil and then to thymine. So, double the number of thymine. So, how to find that which is original thymine and which is converted thymine? How to find that? So, you have to have reference sequence where you have not given bisulfite treatment, and then you compare and then see this in reference sequence C is there, but here T is there, means this T was originally cytosine, unmethylated cytosine. So, like that you can find it out, and then you can find that how many cytosines have been converted or how many cytosines are not converted. Not converted means they were methylated cytosine, and that is what we want to know, how many cytosines are methylated. Okay. Now, there are three contexts. What is after cytosine? Like if I ask you ATGC, okay, and again ATGC. So, what is coming after C? A. So, similarly, you have to find out what are the contexts. There are three contexts, CG. If immediately after CG is coming, that is called CG or CPG, that context is CPG. If after C instead of G any other base is coming, then third one is G. So, C is known, G is known, in between instead of G something else is there, because if G is there, that will immediately come under CG context. If something else is there, that context is called CHG. First is C, in between anything else except C or except G, and third one is G, CHG context. And third situation may be there after C not G, and then third position or G is not there. So, C, H and again H, anything except G is denoted as H, okay. CHH context. So, you have to find out what is the context of methylation, okay. And this has come in the background of, you must be knowing retrotransposons, the jumping genes which were always jumping within the genome. So, that transposons are very much, means methylation, if you find CHG and CHH context, methylation in CG context in transposons are very less. In case of human being, maximum methylation you will find in CG context, okay. But in case of plant you can find everywhere, and we are finding that in case of plant more methylation in CHG and CHH context, okay. Okay, let us go ahead. So, let us see what are the roles, not of course not all, what are the roles being played by epigenetic modifications. Genome stability and defense mechanism, and just I mentioned that transposable elements. You can just imagine transposable elements are nothing like one kid entering into this classroom. What he will start doing, or she will start, just 3-4 years old kid, he will sit on this, means jump on this bench, then he will jump to that, and he will disturb the whole class, isn't it? So, similarly transposons, what they are doing, they are jumping in the genome here and there and disturbing everything. Wherever transposons are going to insert in a gene, that gene is not active, not functional. So, they will disturb the genome. So, somebody should be there, then some instructor will come, or mother will come of that kid, she will take that guy out. So, normal class will be there. So, similarly somebody should be there who should stop the transposable element to not to jump too much, little bit jumping will do. If a little bit bigger guy is there, he will see, oh, so many students, he will be shy, he will not move, he will sit quiet, so something like that, no. So, somebody should be there to ask, or maybe mother comes, saying, beta, do not make noise, you sit quiet, then he may understand and he may sit quiet. So, similarly who will ask transposable element to keep quiet or do not move so much, little bit movement will do, or then he may not jump on that, he may just go on moving in this. So, we will not be that much disturbed, no. So, similarly, so later we found that this methylation, transposable, silent transposable element, the transposable elements which are not jumping, they are highly methylated. Whenever it is getting demethylated, it will start jumping. So, this methylation is controlling jumping activity of the transposable element. So, genomes are having, means try to methylate transposable element, so that stability of genome over the generation. If this is not there, then we know that rice is a rice for last billions of years. Why it has not converted into wheat? Wheat is still wheat, it is not, but little bit evolutionary changes are happening. That is why this variety high yielding, this variety low yielding, this is tolerant, this is susceptible, all those things are there. So, little bit changes are there, but not that much change. Rice will convert into sorghum and maize will convert into pearl millet, it is not like that. So, that is how control is there, genome is stable. Genome imprinting. If you know what is genome imprinting, I will tell you a little bit about, but our target is to give the importance of epigenetic changes and genome imprinting. you know that endosperm, okay, and 3 set of chromosomes are there, 2 set coming from mother and 1 set coming from father. Okay. And if you see, either of the genome are expressing, either mothers or fathers, but not both. One of them is kept silent. Okay. So, if expression is from mother genome or genes expressing from mother genome, that is called female imprinting, and if it is the genes are expressing from male part, that is called male imprinting. Okay. So, other component of the genome, that is heavily methylated to silence that. So, that is going to play in imprinting of the genome. Epigenetic changes are playing role. And you also know that some genome has been suppressed by methylation, but it has not, means endosperm is not going to contribute to the next generation. So, whether it is silenced or not, it is not going to matter next generation. Wherever next generation, whatever genetic material is going to the next generation, that is not affected by methylation or demethylation. It does not mean there is no such changes, that changes are there, but at least from the endosperm that is not happening. Either plant while germinating it will eat, or we are going to eat the endosperm, isn't it, in grains. So, they are not playing important role in transfer of the genetic material. Okay. Adaptation to the environmental stress. So, either in case of animal system or plant system, we find that if plant is under heat stress or drought stress, certain genes required to be more expressed and other genes required to be less expressed. Say for example, drought stress in rice. Okay. If drought stress in rice is there, what is the requirement? Stomata should be closed, so that loss of water because of transpiration should be minimized. Okay. Certain other genes like proline content should increase, so that it can hold water. Okay. So, they are all regulated through epigenetic changes. So, that sort of evidences are being becoming available, and it is going to controlled through epigenetic changes. So, we can also do that, isn't it? If that is happening naturally, if we are going to increase the expression of gene which is responsible for synthesis of proline or closing of stomata, and we have seen no inducible. So, you spray something, it will getting induced and stomata are closed. So, something like that. So, that is where we will see that epigenome editing will be required. But before going into the epigenome editing, we need to identify such marks where to do editing. So, this particular piece of DNA is responsible. If you methylate it, more proline will be produced. If you methylate this, stomata will be closed. So, you have to identify. If you identify, then you can, okay, my target is this one, I want to methylate or demethylate. If you want to methylate, put the methylase transferase. If you want to demethylate, put that demethylase enzyme, and you have to synthesize guide RNA. So, guide RNA will go and bind, and it will allow that enzyme to do its action. Tissue specific expression. Okay. Sometime we want that this particular protein should express more in seeds, or this particular protein should express more in leaf, or that particular gene should express more in root. Okay. So, you have to methylate or demethylate that particular gene in that particular tissue, because expression is being naturally, we know that nature is managing expression of gene in different parts of the or different tissue of the plant through methylation or demethylation. So, we can use that technique to overexpress or suppress the expression of gene. So, that is all for today. If you have any query. mRNA? Okay. See, the role of miRNA is to chop the transcript or to cut the transcript. Say for example, that transcript is of 1 kb. If you make 2 pieces,